macroscopic morphology ठीक है the macro growth pattern on the artificial media size, texture, pigmentation, bacterial colonies, typical size and texture microscopic morphology size cell endoscopic inclusion cellular and contagious so only the microscope this is not a common idea ठीक है स्टेमिंग कर देते हैं ग्राम स्टेमिंग differential integration ठीक है stemming in connection with microscopic morphology environmental अच्छा to grow at a various temperatures in the presence of oxygen and other gases at various pH level in the presence of the other ions and the salts resins अच्छा मतलब किस टेम्परेचर पे ग्रो करेगा ऑक्सीजन का प्रेजेंस रिक्वायर्ड है कि नहीं कितने कितने pH पे ग्रो करेगा आयंस के नीड सॉल्ट्स समझे used various carbon and nitrogen sources as nutritional substrate when grown under a specific environmental condition to nutritional requirement तो यहाँ पे nutritional requirement क्या क्या होगा उसके बाद resistance profiling division inherent resistance to specific antibodies elementals antitoxins antigenic property antigenic अच्छा serological immunological method to determine fitness of mechanism of treatment what are these content curated content families international track India for example and the DC content is not closer to the music content on the track and also the DC content of 50% is not present in chat and DC का 50 है और किसी का DC जो है वो 25 है वो closely related जैसे DNA RNA बेस सीक्वेंसेस कैरेक्टरिस्टिक्स इंक्लूडिंग बेस जनरेशन है, ठीक है। Extant pivot sequence of analysis symmetric sequences. As a whole by hybridization assay. Hybridization assay. ANI. Hybrids nucleotide identification. This method allows multiple coding sequences in microorganisms genome to determine the result of genome sequencing. Computer algorithms. The readiness of the microorganism is very better. Which ठीक है। Genome to genome distance. This is a computerized calculation that uses the difference in silico genome comparison. In silico. With the molecular technique genome to genome distance threshold is 70 nanometer. 70% computer होना चाहिए genome to genome distance. बयालीस मिनट और है भाई, ज़्यादा। Is a method for organisms making groups attacks based on some zoology, genetic traits. The hierarchical classification system consists of the following taxa. डोमेन्स पता है, किंगडम पता है, फाइलम, ठीक है, डिवीज़न होता है वहाँ पे, ठीक है, क्लास, ऑर्डर, फैमिली, जीनस, स्पीसीज, बैक्टीरिया या प्रोकैरियोट्स या प्री न्यूक्लियस, इसको क्या बोल सकते हैं, प्री न्यूक्लियस और द सेपरेटेड इंटू टू डोमेन्स, बैक्टीरिया एंड द आर्किया, दैट इज द एंशिएंट बैक्टीरिया, ठीक है। द बैक्टीरिया कंटेनिंग द इन्वायरमेंटल प्रोकैरियोट्स, द बैक्टीरिया कंटेनिंग द इन्वायरमेंटल प्रोकैरियोट्स, द ब्लूमिंग साइनोबैक्टीरिया एंड द हेटेरोट्रॉफिक मेडिकली रिलेवेंट बैक्टीरिया। प्रोकैरियोट्स मतलब जो बैक्टीरिया होते हैं उसमें मोनोट्रॉफ्स होते हैं, हेटेरोट्रॉफ्स होते हैं। मोनोट्रॉफ्स में भी नहीं आती हैं क्योंकि ये फोटोट्रॉफ्स हैं, कुछ कीमोट्रॉफ्स भी होती हैं, और साथ में हमारे पास यहाँ पे हेटेरोट्रॉफ्स जो कि मेडिकली इंपॉर्टेंट और रेलेवेंट बैक्टीरिया हैं। बाकी अगर नॉर्मल आइसोलेट्स दैट लिव इन एक्सट्रीम हैबिटेट्स, ठीक is a high salt concentration, jet fuels, high temperature. The third domain, eukarya, that is eukaryotes, to nucleus, also contain medical relevant organisms including fungi and parasites. Below the seven level taxonomic sub-levels, seven level taxonomic sub-levels in understand and introducing automatic family. A family encompasses the chromosomes that can contain multiple genera and consists of the organisms with common attribute. The name of the family is found by the suffix -ec to the root name of the object genera. Genera is called the type genus. For example, the Streptococcaceae family. Type genus is Streptococcus. Okay. Streptococcus, Streptococcaceae. One exception to the rule in the microbiomes is Enterobacteriales, is named after the enteric bacteria in the type species in the Escherichia coli. ठीक है, Escherichia coli जो कि टाइप स्पीसीज है उसके बाद तुम्हें देखिए enteric ग्रुप पे देखिए Enterobacteriales bacteria. The species listed in the according to the values of the International Committee for the Systematic Prokaryotes. International Committee for the Systematics in the Prokaryotes in the International Code of the Nomenclature of Prokaryotes. International Code of Nomenclature of the Prokaryotes में दिया International Committee for the Systematics of the Prokaryotes. ठीक है. Type species of this term. This could run the guideline for geology, the nomenclature, classification, categorization, gender, physiological, biochemical, genetic, and the phenotypic traits of the organism. Okay, okay. Now the microorganism type species will be described in detail using the diagnostic and the comparable methods. So and authentic instance must be available for the future analysis. Okay. Genus taxonomy containing different species that have a single important feature in common. Species with the genus differ sufficient to maintain its status as an individual species. Placement of a species within a particular genus is based on the various genetic and the phenotypic characteristics found among the species. Microorganisms possess the multitude of the physical feature exhibited by the higher organisms such as plants and animals. For instance, there are leaf and fossil record. Exhibit a tremendous capacity to intermix genetic material among the seemingly unrelated species in the genera. For these reasons, confidently establishing a microorganism's uniqueness in higher taxa beyond the genus level is difficult. All the grouping similar genera into common families and the similar families into the common order used for classification of the plants or animals. Hand designations, division, class, and the order are not useful for classification of bacteria. Species singular is the most basic of the taxonomic group and can manage a collection of the bacterial stem. It is a bacterial stem collection that has the common physiological and genetic features and differ notably from other microbial species. Classical taxonomic subgroups within a species called the subspecies are recognized. Furthermore, designations such as a biotype, serotype, or phenotype may be given to groups below the subspecies. Subspecies के भी नीचे हमें देखने को मिल सकता है क्या, बायोटाइप, सेरोटाइप, फिर जीनोटाइप. For example, Klebsiella pneumoniae and the Klebsiella oxytoca are the two distinct species within the genus Klebsiella. ठीक है, बोल सकते हैं. Serratia odorifera biotype 2 and the Treponema pallidum subspecies pallidum are examples of the biotype. Biotype is considered the same species with the same genetic makeup but display different physiological characteristic. Subspecies do not display significant enough divergence to be classified as a biotype or new species. Although these subgroups may have some taxonomic importance, the usefulness and diagnostic advantage. ICMP, that is International Code of Nomenclature of the Prokaryotes. International Code for Nomenclature of the Prokaryotes. It provides the accepted level by which organisms are universally recognized because genus and the species are because genus and species are the groups of the common listed by microbiologist. The discussion of the rules governing microbial nomenclature is limited to these two texts. The binomial two-name system of nomenclature, every organism is assigned a genus and a species based on the great variation. Each organism has a scientific label consisting of two parts: genus designation, in which the first letter is always capitalized, and the specific designation, in which the first letter is always lowercase. The two components are used simultaneously and are presented in Linux order and Latin scripts. For example, the Streptococci, Streptococcus pneumoniae, Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus bovis, Streptococcus bovis, agalactiae, pyogenes, and pneumonia. Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus pneumonia, Streptococcus bovis, among others. Alternatively, the name may be abbreviated by using the uppercase from the first letter of the genus designation followed by a period and the full species name. Okay, next is the Streptococcus हो गया, pneumonia, Streptococcus agalactiae and the bovis. Finally, when discussing the single species organisms, the species may be designated using a sp dot and a group of species with the genus. Group of species within the genus species. For example, Streptococcus species and the Streptococcus species. अगर एक की बात कर रहे हैं single specific organism, then there is a Streptococcus species. Okay. Frequently, an informal designation, that is a Staphylococci, Streptococci, and Enterococci, may be used to label a particular group of organism. These designations are not capitalized or declined. Okay. As more information is gathered regarding organisms classification and identification of the particular species may be moved to a different genus or assigned a new genus name. The rules and the criteria for these changes are beyond the scope of the chapter, but such changes are documented in the International Journal of Evolutionary and Evolutionary Microbiology. Like this documentation may be found at it is important to note that the fungi and the parasites are so difficult to maintain and may not reflect the current validity at the time of the GI. In the diagnostic laboratory, changes in the nomenclature or faced gradually, so then the patient laboratorians have the authority to recognize later and the person has been given name. This usually first by using the new genus designations or continue to provide the previous designation. For example, Stenotropomonas, Mantophila, Burkholderia, Pseudomonas, Sebaceae. Microbial identification is the process by which the microorganisms key feature are delineated. Once these features have been established, the profile is coupled with those who were previously characterized microorganisms. Organisms can then be assigned to the most appropriate taxon, can be given appropriate genus and the species name. These are essential aspects of the taxonomy in diagnostic microbiology and the management of the infectious disease. Identification method. A wide variety of the method and the criteria are used to establish a microorganism's identity. These methods can be separated into either of the two general categories: genetic and phenotypic characteristics. Genetic characteristics due to the organism's genetic makeup, phenotypic including the nature of the organism's gene, nucleic acids. For more information about the microgenetics. Phenotypic characteristics are based on the features beyond the genetics level between the both usually observable characters from the features that may require extensive analytic procedures to be detected. Example of the characteristics used as criteria for the classification and the classification of the provided in modern microbial taxonomy use a combination of the several methods to characterize microorganisms thoroughly to classify and the name each organism. All the criteria and the examples are given in the context of microbial identification for classification purposes. The principle and the practices of the classification framework, the approaches used in the diagnostic microbiology for the identification and the characterization of the microorganism encounters in the clinical setting. Because of previously effort and the accomplishment of the microbial taxonomist, microbiologists do not have to use several programs and classification and identification scheme to identify infectious agents. Instead, microbiologists use key phenotypic and genetic features on which to base their identification to provide the chemically relevant information in a timely manner. This should not be taken to mean that the identification of all clinically relevant organisms is easy and straightforward. This is also not mean to imply that microbiologists can identify and recognize only that have already been characterized and named by taxonomists. Indeed, the clinical microbiologist laboratory is well recognizable as a place where the previously unknown or the uncharacterized infection agents are initially encountered, and as such it has an even increasing responsibilities to be the source of information and the reporting for the emerging etiologies of the infectious disease. Role of taxonomy in the diagnostic microbiology stabilizes and maintains the records of the key characters of the clinically relevant microorganisms, facilitates communications among the technologists, microbiologists, physicians, scientists by assigning universal name to clinically relevant microorganisms. This is establishing an association of the particular disease or the syndromes with specific microorganisms, epidemiology, and the tracking outbreaks. Accumulating knowledge regarding the management and the outcome of the disease associated with the specific microorganisms, establishing pattern of resistance to the antimicrobial agents and the recognition of the changing microbial resistance pattern, understanding the mechanism of antimicrobial resistance and detecting new resistance mechanism exhibiting by the microorganism, recognizing new and the emerging pathogens. Recognizing changes in the type of the infection or the disease caused by the characteristic microorganisms, revising or updating the available technologies for the development of the new method to optimize the detection and the identification of the infectious agent and the detection of the tends to the anti-infective agents, microbial, viral, fungal, and the parasitics. Development new anti-infective therapies, microbial, viral, fungal, and the parasitic. The most specific and the exclusive taxon used in Alzheimer's disease. The process consisting of a series of methods designed to provide the microbiologist with the relevant and the useful clinical information about the microbiome is taxonomy. Classification and the naming of the organisms are useful in diagnostic microbiology. All the following except providing a standardized grouping. Which of the following is not correct? Is it the paranormal one? Is it the Staphylococcus aureus? Staphylococcus aureus, correct. This is the structure and the organization of prokaryotic chromosomes, including the number, relative sizes, and the cellular locations. Outline the basic processes and the essential components required for information transfer and replication, transcription, translation, and the regulatory mechanism. Define the mutation, recombination, transduction, transformation, and the conjugation. Describe how genetic alterations mechanism for the evolutionary and the survival organisms. Differentiate environmental oxygenation and the final electron acceptor. Differentiate environmental oxygenation, final electron acceptor, aerobes, facultative anaerobes, and the strict anaerobes in the formation of energy. Compare and contrast the key structure elements, cellular organizations, and the types of the organism classified as a prokaryotic and the eukaryotic. State the functions of the biological significance of the following cellular structures: the outer membrane, cell wall, periplasmic spaces, cytoplasmic membrane, capsule, fimbriae, pili, flagella, nucleoid, and the cytoplasm. Difference in the organization and the chemical composition of the cell. Look for gram-positive and the gram-negative bacteria. Microbial genetics, metabolism, and the structure. Genetics, metabolism, and structure are the key to the microbiology and survival. This process involves a numerous pathways that are the widely often complicated and frequently interactive. Essentially, survival requires nutrients and energy to fuel the sensitive material necessary to grow, propagate, and carry out the other metabolic process. Survival requires the nutrients and energy to fuel the sensitive material needed to grow, propagate, and carry out the Although the goal of survival is the same for all organisms, the disease mechanisms uses to accomplish this varies substantially. Knowledge regarding the genetic, metabolic, and the structural characteristics What is the genetic, metabolic, and the structural characteristics of the microorganisms provide the basis for the understanding almost every aspect of the diagnostic microbiology, including the mechanism by which microorganisms cause disease. Okay. The development and the implementation of the techniques for microbial reduction, cultivation, identification, and the characterization. Okay. Detection, cultivation, identification, and the characterization. Antimicrobial action and the resistances. The development and the implementation of the test for the reduction of the antimicrobial resistance. Potential strategies for disease therapy and the control of the potential strategies for disease therapy and the control. Microorganisms vary significantly in their genomic and the metabolic pathway. Genomic and the metabolic pathways, therefore structure varies significantly the genomic and the metabolic pathway, and therefore the structure. A real consideration of these differences is beyond the scope of this textbook. Therefore, a generalized description of the bacterial system is used. Generalized description of the bacterial system is used as a method to discuss microbial physiology and the structure. Information regarding characteristics of the fungi, parasite, and the virus can be found in the subsequent chapter for each specific taxonomic group. Bacterial genetics. Genetics, the process of the heredity and the variation. What is genetics? It is a process of the heredity and the variations. It is the starting point from which all other cellular pathway functions and all other cellular pathway functions and structures originated. The ability of a microorganism to maintain the viability, adapt, multiply, and the cause disease is driven by the organism's genetic compositions. Maintain its viability, adaptability, and the multiplicability causes disease. The three major aspects of the microbial genetics that require discussion included the structure and the organization, replication, expression of the genetic information, mechanism by which genetic information is altered, and the exchange among the bacteria. Now the energy and the nutrients are required for biosynthesis, assembly of the cell structure, waste removal, motion and the other responses to the environment, genetic process. It is a bacterial cell. Nucleic acid structure and the organization. For all living genes, energy information resides or is encoded in nucleic acid. The two major classes of nucleic acid are the DNA and the most which is the most common macromolecules that encode genetic information is ribonucleic acid. In some form, RNA encodes genetic information for the various viruses and other forms. RNA plays an essential role in the several of the genetic processes in the product of the eukaryotic cell, including the regulation and the transfer of information. Prokaryotes or the prenuclear organisms do not have a membrane model organelles and the cells. Genetic material is therefore not enclosed in the nucleus. In favor of the nucleus, organisms have the genetic material is enclosed in a nuclear envelope. Nucleotide structure and the sequences. DNA consists of the deoxyribose sugar captured by the phosphodiester bond and the bases that are covalently linked. Bases linked to each of deoxyribose sugar are the key to the genetic code within the DNA molecules. इसका मतलब डीऑक्सीराइबोस शुगर जो होता है वो फॉस्फोडाइस्टर बॉन्ड से जुड़ा होता है फॉस्फेट से डीऑक्सीराइबोस शुगर connected by phosphodiester bond the bases that are covalently linked to each deoxyribose sugar within the DNA. The four nitrogen bases include the purines, adenine, and the guanine, pyrimidine, cytosine, and methionine. The amino acid replaces the combined sugar, phosphate, and the base. Sugar, phosphate, and the base. That is the nucleotide. ATP, that is the adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, thymine triphosphate, purine triphosphate. These are all the nucleotides. These are all the example of the nucleotides. Adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, and the thymine triphosphate, uracil triphosphate. You didn't. Given are the nucleotides polymers. Nucleotide polymers chain of the strand and the order of the bases around the DNA understanding of the base sequences. The sequences provide the information that codes for the protein, that will be inside by the microbial cell. This is a sequence, that is the sequence in the genetic code. One minute. Phosphodiester bond. Phosphate, 3' hydroxyl, 3' hydroxyl, 5' phosphate, phosphodiester bond. Ester. Ester अच्छा होता है ना, ठीक है. Deoxyribonucleic molecular structure, the intact and the molecular active nuclear polymer strand of the 5' phosphate hydroxyl terminus, which is an anti-carbon dependent master process of the strand and also complementary. Complementary, the adherents of the ATGC display intervention of DNA, double helix, but two anti-carbon single stranded DNA from twisted level structures. In addition, the replicated base pair provide the format for consistent replication and the expression of the genetic code. In contrast to DNA, which carries the genetic code rarely exists as double stranded molecule, rarely exists. There are the four major types of RNA: messenger RNA, transfer RNA, ribosomal RNA. ठीक है, 4 मेजर टाइप ना, तो मैसेंजर हो गया, ट्रांसफ़र हो गया, राइबोसोमल हो गया, अलॉन्ग विद वैरायटी ऑफ नॉन कोडिंग आरएनए है ना, एनसी आरएनए. मतलब हमारे पास 4 टाइप का आरएनए होता है ना. एक हो गया मैसेंजर आरएनए, ट्रांसफ़र आरएनए, राइबोसोमल आरएनए और नॉन कोडिंग आरएनए. ओके. Molecules such as the microRNA that play a key role in the post-transcriptional regulation of gene expression and a post-transcriptional regulation of gene expression. Gene and the genetic code. A DNA sequences that encodes a specific product, RNA or the protein, is defined as a gene. A DNA sequence that encodes a specific product, चाहे वो RNA बोलो चाहे protein बोलो, is defined as a gene. Thousands of genes in an organism encode messages or the blueprints for the production of one or the more proteins and RNA products that play essential metabolic role in the cell. All the genes in an organism comprises the organism's genome. The genome of a microorganism includes the chromosome and the mobiliosome. All the genes in an organism comprise the genome. The genome of a microorganism includes the chromosomes and the mobilomes. Mobilome, extra chromosome. The size of the gene and the entire genomes are usually expressed in number of base pair. किलो बेस पेयर में 103 बेसिस होते हैं, मेगा बेस पेयर में 106 बेसिस होते हैं. किलो बेस पेयर में 103 होते हैं, मेगा बेस पेयर में 106 होते हैं. So these are the widely distributed various organisms for the species. In addition, the base pair sequences for individual gene may be highly conserved. So dented sequence difference among the different organisms. this correlates to the age, asymmetry, and the differences in the content in the sequence of the visual monitor method. Is it identifying the factors and acronyms? Okay. The chromosome. Genome is organized into discrete elements known as a chromosome. The set of the gene with in a given chromosome is arranged in a linear fashion, but the number of genes per chromosome is variable. Similarities along with the number of chromosomes per set is consistent for a given species. This number varies considerably among this. For example, humans can replicate the ploidy of the chromosome, however, bacteria containing a single unpaired, that is a haploid chromosome. Number 2, the number of chromosomes. Number of genes per chromosome is variable, so the number of chromosomes per cell is constant. Bacteria classified as a prokaryotes. Therefore, the chromosomes is a number not less. The bacteria chromosome contains the genes that cause viability and release to the double stranded. The bacteria chromosome containing the gene which is about the viability and release to double stranded is closed circular macromolecule. Double stranded closed circular macromolecules. The molecule is extensively folded and twisted, so required to fit within the confined space in the mature cell. The linearized unsupercoiled chromosome of the bacteria E. coli is Escherichia coli is 1 long, but its surface length are 1 to 3 micrometer. The length of the Escherichia coli and pseudocoid chromosome is 130 micrometer long, but it's within 1 to 3 micrometer. This attempts to the extreme compact structure of the supercoiled bacterial chromosome for the genes in the compactor chromosome to be explicitly replicated. Unbinding or the relaxation of the molecules is required. In contrast to the bacterial chromosomes, the chromosomes of parasite and fungi numbers more than the one per cell are linear and are housed within under moderate organisms is linear. This difference is a major criteria for classifying bacteria as the prokaryotes and the fungi and the parasitic eukaryotes. The genetic makeup of a virus may consist of DNA or RNA contained within a protein code present in a cell. Now the non-chromosomal elements, that is a mobile What is mobilome? Mobilomes are the non-chromosomal elements known as a mobilome. Non-chromosomal elements known as a mobilome. Although the bacterial chromosome present the majority of the cell's genome, not all the genes are confined to the chromosome. Many genes may also be located on the plasmids or the transposable elements. Both of these extra chromosomal elements are able to replicate and encode informations for the production of the various cellular products. Many of these elements are replicated by the integration to the host chromosome. Both represented to the host chromosome अच्छा ठीक है not its chromosome host chromosome whereas others referred as the episomes are capable of replicating independently from the host chromosome. Episomes, okay, both of these extra chromosome elements are able to replicate and encode information for the whereas many of these elements replicate by the episomes are capable of खुद से कर लेते हैं क्या. Although considered part of the bacterial genome, they are not as stable as the chromosome. Not as stable as the chromosomes, and they may be lost during cellular replication, often without any detrimental effect on the viability of the cell. Plasmid exists as a double standard closed circular autonomously replicating extra chromosomal genetic element ranging in the size from the 1 to 2 kilobase pair to the 1 megabase pair. The more kilobase pairs 1 to 2 kilobase pair 106, megabase pair is 106. Okay. The number of the plastids per bacterial cell varies extensively, and each plastid is composed of the several genes. Some genes encode the product that mediates the plasmid replication and the transfer between bacterial cell, whereas the other encodes product that provide the specialized functions as the germination of the antimicrobial resistance, the unique metabolic process. Unlike the most chromosomal gene, plasmid genes do not usually encode for a product essential for the viability. Plasmids in whole or in part may also become incorporated into the chromosome. Transposable elements piece of the DNA one genetic element to another from the plasmid to the chromosome, or vice versa. Many other replicate independently and do not exist separate entities in the bacterial cell. The root of the transposable elements in the simple transposome, insertion sequences, and the composite or complex transposome. Simple, complex, composite. Simple is insertion sequences. Insertion sequences are presented to the containing the genes that encodes information required for the movement from the one side in the genome to one side. अच्छा, दो टाइप होते हैं. The insertion sequences are limited to contain the genes that encode the information required for the movement from one side to the genome to the other. Composite transposome are the cassettes. Flanked by the insertion sequence, flanked by The internal gene can live into the yes codes for the plasmids and the transposable elements coexist with the chromosome in the cell of many bacteria species. Plasmids and the transposable elements coexist with the chromosome in the cell. These extrachromosome elements play a role in the exchange of genetic materials for the bacteria, microbiosphere, and organizations among the clinical relevant bacteria. No DNA replication. Bacteria multiply by the band diffusion across of these and resulting in the production of the two daughter cells from one parent cell. As a part of this process, the genome must be replicated. In each daughter cell. This is an identical copy of the functional DNA. Replication is a complex process mediated by the various enzymes of the DNA polymerase cofactors. Replication must occur quickly and accurately. Descriptive purposes, replication may be considered in the first stages for discovery purposes. Unwinding or relaxation of the chromosomes, supercoiled DNA, separation of the two complementary strands of the parent DNA. Each strand may serve as a template, that is patterns for the sense of the new DNA strand, referred to as a semi-conservative replication. Sense of the new DNA strand, termination of the replication, releasing the two identical chromosomes, one for each daughter. एक घंटा तो मेरा पूरा हो गया.